Hey. Welcome to a world of laughter, learning, and entertainment. Greetings, viewers. Get ready to immerse yourself in the world of interleukin-13 receptor in this video. The interleukin-13 receptor is a type I cytokine receptor binding interleukin-13. It consists of two subunits, encoded by IL-RA and IL-4 respectively. These two genes encode the proteins IL and IL. These form a dimer with IL-13 binding to the IL chain and IL stabilise this interaction. This IL-13 receptor can also instigate IL-4 signaling. In both cases this occurs via activation of the Janus kinase JAK signal transducer and activator of transcription STAT pathway, resulting in phosphorylation of STAT. Phosphorylated STAT dimerises and acts as a transcription factor activating many genes, such as eotaxin. There is also another receptor that can bind IL-13, encoded by the IL-RA gene. This binds IL-13 with very high affinity and can therefore sequester it but does not allow IL-4 binding. It acts as a negative regulator of both IL-13 and IL-4, however the mechanism of this is still undetermined. As we progress, let's zoom in on function and examine its role in shaping our overall narrative. Interleukin-13 IL-13 is an effector cytokine partially sharing the signaling pathways with IL-4 due to the utilization of a common receptor system IL-4 receptor type 2. A private receptor system, binding specifically IL-13 with high affinity, seems to use different signaling pathways and is increasingly being studied for its potential as a novel prognostic factor, biomarker or therapeutic target in different types of cancer. As we venture forward, let's take a closer look at the shared IL-4-IL-13 receptor and its impact on our understanding. IL-13 uses the IL-4 receptor type IL, the complex formed by an IL chain and an IL chain. Initially, the ligand IL-4-IL-13 binds to the IL chain and IL respectively. Thereafter, a secondary chain IL and IL respectively will also bind, forming the complete IL. The IL complex, however, can also bind to a different secondary chain, the IL-RA, forming the IL-4 receptor type IL. In non-hematopoietic cells, IL-RA is poorly expressed. On the other hand, IL is poorly expressed in lymphocytes but abundantly in all non-hematopoietic cells. Myeloid cells express both of them to a certain degree. This different distribution of secondary chains accounts for the difference in distribution of completed receptors, with being IL prevalently expressed in lymphocyte and IL prevalently in non-hematopoietic cells. Consequently, only IL-4, through IL, is able to modulate the function of lymphocytes, inducing polarization and B cells IgG class switching, while IL-13 is mainly acting on myeloid cells and non-hematopoietic cells, having strong effects on mucus production, smooth muscle contraction, epithelium permeabilization, e.g. allergic asthma. After the complete assemblage, the conformational changes in IL or IL tails leads to the intracellular signaling, starting with the auto and cross-phosphorylation of associated JAK kinase JAK for IL-RA, JAK for IL, JAK and TIC for IL, and followed by phosphorylation of intracellular domains of IL in critical Y residues, which are therefore activated to form the docking sites for downstream signaling molecules endowed with SH domains. While the docking sites in IL and consequently IL-4 are able to efficiently activate both STAT and AS signaling molecules, IL and consequently IL-13 only activates effectively STAT. Activated STAT molecules form dimers which translocate to the nucleus to bind responsive elements, e.g. promoter in B cells, arginase enhancer in macrophages. The binding affinity of IL-4 for IL is much higher than IL-13 for the IL, hence IL-4 would outcompete IL-13 for receptor availability within IL at parity of concentration. Let's now enter the realm of the private IL-13 receptor and discover the fascinating stories it has to tell. Besides IL chain which work in conjunction with the IL, IL-13 can bind with much higher affinity to IL. IL-13 presents 35% homology with IL-13 and it is expressed mostly in structural cells but also has been identified in fibroblasts and, only in mice, in soluble form. It presents an extraordinary affinity to IL-13 but does not form complexes with any secondary chain. Because of the apparent lack of signaling domain and the short tail, it has been initially thought not to have any signaling activity, and regarded as the receptor, that is its function would just consist in competing for IL-13 binding and neutralizing his effect. Indeed, it has been shown that IL-13 blocks IL-13-driven STAT signaling by binding IL-13 with high affinity. However, partial block is also extending to IL-4-driven STAT signaling, presumably due to the cytoplasmic domain interfering with the assembling of IL-13 with a secondary chain. However, increasing evidences are accumulating that IL-13 is more than a decoy. IL-13 signaling through IL-13 and up-driven TGF production has been initially reported in monocytes and then confirmed in mouse models. According to these studies, IL-13, through the overexpression TNF-induced of IL-13 would be able to activate AP1 signaling and production of TGF, driving pro-fibrotic effects. Some recent works is evidencing how wide range of signals can be actually activated by this receptor, e.g. undetermined, MABCIC, ACTVIC, PIPC in normal or pathologic environments. How IL-13 might overcome the limitation of a 17 amino acids short tail lacking any signaling motif, it is not clear yet, but it has been shown that, at least in some cases, the association with other receptors or signaling adapters can do the trick. Your feedback helps me improve, so please take a moment to leave a comment or review.